Okay everyone, welcome back to our next lecture video. This video is going to cover the blood vessels, which is our final component of the cardiovascular system. So up until this point, we've covered 2/3 of the cardiovascular system. We've covered the blood, we've also covered the heart, and in this presentation we're going to finish up this system with the blood vessels. Now the blood vessels are so extremely important because they are going to connect the two different sides of the heart and allow for blood flow throughout the body. Blood is going to travel through our blood vessels, and that's going to allow for the transport of oxygen out to our body tissues. We're going to finally break down the different types of blood vessels and each of their purposes, as well as a concept known as hemodynamics, which is referring to the physics of how our blood is going to travel in the blood vessels. So as we continue to move forward with this system, just like the rest of the systems that we learn in this class, we want to understand the structure of the system, and that will give us some intuition about the function. So the first thing that I really want to cover with blood vessels is their structure. Now there are lots of different types of blood vessels, and each type of blood vessel is going to have different characteristics. However, one way to organize blood vessels are by the layers that make up the walls of the blood vessels, and those three layers are listed on this slide. Each type of vessels are going to have their own individual differences. Some vessels will only have one of these layers. Some vessels will have larger versions of these layers. And in other vessels, we'll see that these layers are going to drastically differ between two different types of vessels. So let's dive into the three major layers of the blood vessels, which are the tunica intima, the tunica media, and the tunica externa. The tunica intima is also often referred to as the tunica interna, and it is called this because it is the innermost layer of all of our vessels. Every single blood vessel in our entire body will contain this tunica intima. This will be the one layer that is continuous through all of our vessels. So this innermost layer is made up of a couple other layers, namely an endothelium, as well as the elastic tissue that sits outside of the endothelium. So every blood vessel is going to have a lumen where blood is going to be found, and that lumen is lined by this endothelial tissue. This is going to be the layer that contacts the blood inside of the vessels, literally. Now the next layer is the tunica media. I like to remember the tunica media, it starts with an M, is going to be our middle layer. And I guess I didn't say this with the tunica intima, but the tunica intima that starts with an I is our innermost layer. So the tunica media has two major characteristics that distinguish this layer from the others. The tunica media contains a layer of smooth muscles that are circularly arranged, and it will also contain additional elastic fibers. We will find an external elastic lamina making up part of the tunica media. Now, both the smooth muscle and the elastic fibers are going to give the specific characteristics that help us to differentiate blood vessels. Some vessels are going to have very, very large layers of smooth muscle, and that's going to help to regulate blood flow. And other layers are going to have lots and lots of elastic fibers, and that's going to increase the amount of stretch that our vessels are allowed to feel. The amount of smooth muscle and the amount of elastic fibers will change depending on the vessel we are looking at. However, most of our vessels will contain the tunica media. Now the tunica externa is the outermost of all of the layers. I like to remember that this is the most external layer. There will be some elastic tissue in this layer, but there's also primarily going to be found collagen fibers making up part of the tunica externa. Blood vessels and nerves are going to travel in the tunica externa, and these will actually supply the walls of the vessels themselves. We don't often think that our blood vessels themselves need to be perfused, but in fact they do. So, therefore, we have very, very tiny vessels that are going to supply the actual tissues of the vessels themselves. Those smaller vessels are called vasa vasorum, and this means the vessels to the vessels. This connective tissue layer wrapping around the outside of our vessels is going to be integral for keeping our vessels in the right places and helping anchor the vessels to the surrounding tissues. So knowing these three layers that can be found in our blood vessels are going to get you started on understanding the different types of blood vessels that we have and the differences that can be found between them. So we will come to see that the makeup of each of these different layers of our blood vessels will change depending on the vessels, but we want to look at our blood vessels in terms of these layers as we start to learn the different types. So here we can see a couple different blood vessels laid out for us. We can see here, this is an artery, here we have a vein, and over here we have a capillary. Now, one thing that we really want to take a second to remember is that arteries specifically will carry blood away from the heart, and veins will specifically carry blood back to the heart. Now, arteries are most often depicted in this reddish color because that's going to indicate that arteries carry our oxygenated blood, and veins are going to be depicted in this bluish color, indicating that veins are going to carry deoxygenated blood. There is one exception of this, and those are the pulmonary arteries and the pulmonary veins. The pulmonary arteries are the one set of arteries that are often depicted in blue in our body, and the pulmonary veins are the one set of veins that are often depicted in red. Because, in fact, after the pulmonary circulation, the pulmonary veins will take oxygenated blood back to the heart. So those are one major difference in terms of our understanding of arteries and veins being red and blue. But now that that is out of the way, we're really focusing on the systemic arteries and veins, and that's why this artery is depicted in red and this vein is depicted in blue. Now, what we can start to see from this slide is that arteries and veins are going to have some of the same layers, and they will also have quite a few similarities. However, there are some major differences between the general structures of arteries and veins. So you can see each of these layers of our blood vessels, which we just discussed, can be found in both arteries and in veins. But what you may notice is that these layers will represent differently in different vessels. So let's go ahead and get started with the tunica intima, or the tunica interna, as we can also call it. The tunica intima has three major sublayers that make it up. First and foremost, we'll find the endothelium, we'll find the basement membrane, and the internal elastic lamina are three major layers making up the tunica intima. Now, endothelium is defined as the epithelial tissue that lines the inside of our blood vessels. If you ever hear endothelium, you want to think about our arteries and our veins. All blood vessels will be lined by this endothelium. And this, in fact, the endothelium is going to be the layer of the tunica intima that is present in every single blood vessel. There are other blood vessels that will not contain any of these other layers, but what is always true about blood vessels is there will be an endothelium. And in fact, the endothelium is what is continuous with the endocardium that's lining the inside of the cardiac chambers. So this is our epithelial tissue that lines the insides of the vessels. This is a simple squamous epithelium, and that permits for diffusion going through these cells. Now, certain vessels will allow for diffusion and gas exchange, others won't, and I'll discuss that in just a short while. But what we also will find in the tunica intima is a basement membrane. Now, basement membranes are always associated with epithelial tissue, so naturally, if we're going to have an endothelium, we have to have a basement membrane as well. The other major layer of the tunica intima is the internal elastic lamina. And because there's an internal elastic lamina, you'll see over here that there will also be an external elastic lamina that's found as part of the tunica media. And we'll get to that in just a second. But the internal elastic lamina can be found on the outside of the basement membrane, and it's the outermost layer of the tunica intima. The internal elastic lamina has these little pores that can be found in it that make it look like a slice of Swiss cheese. These pores facilitate diffusion of certain substances from the tunica intima into the thicker tunica media. So the next layer is the tunica media, and the major players that make up this layer are the smooth muscle that is circularly arranged and the external elastic lamina. Now, circularly arranged smooth muscle is a very important statement that I have just made. Certain muscular layers can also be arranged longitudinally, and we'll see those a lot in the digestive system, but circularly arranged muscles are shown here wrapping around the vessel. Just as a ring wraps around your finger, circular muscles will wrap around the outside of this layer of the tunica intima. So the necessity of understanding that these are circularly arranged muscles is that muscles that are arranged in a circle, when they constrict, will decrease the diameter, and when they relax, that's going to increase the diameter of the blood vessel. So it is the smooth muscle in the tunica media that is responsible for the phenomenon of vasoconstriction and vasodilation, which are concepts that we will talk about a little bit later. We will find smooth muscle in both arteries and in veins. And in fact, in all of these layers that we find, the only ones we don't find in the veins are the internal and external elastic lamina. Veins are going to have all of the same layers that our arteries are going to have, but they will be arranged differently, and I'll address these shortly. But for now, to finish up the tunica media, we will also have the external elastic lamina, which is going to give more elasticity to our vessels. And lastly, the tunica externa, which sits out here, is primarily going to be made up of other elastic fibers and collagen fibers, and this is going to anchor our vessels to the tissue that is surrounding. Now, one thing that you can notice just grossly by looking at the arteries and the veins is that arteries are going to have very thick walls, and veins are not going to have as thick walls. Veins' walls are going to be much thinner than the walls of the arteries. And this is because arteries will have a much thicker tunica media, and they're also going to have these internal and the external elastic lamina. Arteries will naturally be a system that contain more pressure. Since blood is getting directly pumped into the systemic arteries, those vessels are going to have a much higher pressure in them than the veins will have. And it is because of this high pressure that we must have very sturdy and very thick walls. So it is the elastic tissue in the walls that help for the stretch of the arteries, and since the veins are a much lower pressured system, they're not going to need as much of a thick wall as the arteries. We will find that the tunica intima and the tunica media inside of our veins tends to be a lot thinner. You can see specifically the tunica media here in our arteries, and it's much thicker than that of in the veins. But veins will have a structure that our arteries will not. And I am referring to the valves. Veins will have valves, and that is because there is a much lower pressure in these vessels compared to the arteries. In fact, sometimes the pressure is so low in our systemic veins that blood actually wants to flow backwards, and that is why we need to have these valves. Just as we had valves in the heart to prevent backflow, we'll have them in the veins to prevent backflow as well. These valves are arranged so that when blood is squeezed in the correct direction, those valves will open up and allow blood to continue flowing, and as that blood tries to flow backwards, that's going to close the valves and prevent any backflow from taking place. Now, the other vessel that we find on this slide is known as the capillary. I've mentioned capillaries a few times as we talked about the pulmonary capillaries and the systemic capillaries when going through the pathway that blood takes as it goes through the heart. So we're going to more specifically elaborate on capillaries, what characteristics they have, and what actions they facilitate. So capillaries, in terms of structure, are the most simple of all of the vessels. Capillaries are a one-cell layer thick type structure that's going to be made up of the single layer of endothelium and the basement membrane that endothelium sits upon. Capillaries are the smallest of all of the vessels in our bodies. The diameter of capillaries ranges from 5 to 10 micrometers, which is extremely small. Since we can only see capillaries with the microscope, we refer to our body's capillaries as the microcirculation. In total, our bodies have approximately 20 billion capillaries. It is in the capillaries specifically that we find gas exchange taking place. Because capillaries have very thin walls, specifically one cell layer of simple squamous epithelium, this is the perfect environment to facilitate exchange of oxygen and carbon dioxide. So it is in the capillaries and the capillaries only that we will have gas exchange taking place. So based on the structure of these three vessels, we can really tell quite a lot about each of these vessels. Arteries are going to have this very thick tunica media that's going to regulate blood flow in terms of vasoconstriction and vasodilation. The thick walls are also going to contain two different layers of the elastic lamina, And that's going to allow for these vessels to stretch due to the high pressure in the arterial tree. Veins are going to have a much wider lumen, but they're going to have much thinner walls. Since veins have a much lower pressure within them, they have to have valves so that they can prevent backflow of blood since the pressure is not as great. And the capillaries, being a single cell layer thick, are going to facilitate oxygen and carbon dioxide exchange with the tissues. Now we just discussed arteries, capillaries, and veins. However, those are not the only types of vessels that we have in the body. We have two other very small vessels, namely the arterioles and the venules. We've discussed that arteries take blood away from the heart and veins will bring blood back to the heart. These are two of the largest of the vessels that we have. Arteries, by definition, are very, very big. But as the aorta leaves the heart, many different branches are going to come off of it, and those are the arteries. Arteries are going to eventually get smaller and smaller and smaller as they continue branching and traveling systemically throughout the body. And as they get smaller and smaller, they're going to eventually be called arterioles, which are some of the smallest arteries in the body. Arterioles are directly going to feed into different capillary beds, and that's how capillaries are going to exist within, in beds of about 10 to 100 capillaries. Capillaries are much smaller than the arterioles, but the arterioles are considered the smallest arteries. Capillaries are not considered arteries and they're not considered veins. They are considered their own individual type of vessel. This is the location of gas exchange. Now, as for the venules, capillaries are going to connect the arterial tree with the venous tree. So it is through capillaries that arterioles and venules will be connected with each other. The venules are going to drain the newly deoxygenated blood from our capillaries and eventually start to meet up. Venules are going to continually meet up and form larger and larger vessels, which will eventually be called veins. And veins are also eventually going to continue to fuse with one another until they get large enough that we form the superior and inferior vena cava that will eventually take blood back into the right atrium. So these are our 5 different types of vessels, and each one of these vessels is going to have its own individual structure. Just as we saw on the last slide, arteries, capillaries, and veins all look different. Arterioles are going to look similar to arteries, but they won't be exactly the same as what we saw on the last slide, and the same goes for the venules. You can see this picture over here is depicting the heart with the systemic circulation. As arteries get smaller and smaller and smaller and continue to branch off, we're eventually going to form arterioles, which will eventually lead into capillary beds, and a capillary bed is shown right here. These capillary beds are going to connect the arterial tree with the venous drainage, and the venules are going to take that deoxygenated blood with them. They're going to continue to fuse with other venules until they get larger and larger and eventually form veins that will bring that blood back to the heart. Now, the last thing that I want to share with you all about this slide is that our vasculature, our cardiovascular system, is a closed transport system. What this means is that there's no entry point into the cardiovascular system. There's a fixed amount of volume inside of this closed cavity. Now, as these vessels are named, this is a very systemic system. There's quite a lot of space that's found inside of our vessels. However, we have to keep in mind that if things are going to get in and out of our cardiovascular system, we either need to do that artificially with one of our medical tools, or perhaps we'd have to use capillaries. But capillaries don't have any explicit opening, so it's strictly based on diffusion that things get in and out of capillaries. So this system is completely closed off to the external environment. Certain structures are contained inside of this system, and completely different structures will be found on the outside of this system. We discussed in the last recording that our body contains about 5 to 6 liters of blood. So naturally, we can deduce that there's about 5 to 6 liters of space or volume inside of this closed transport system. And if we were to increase our blood volume, that's going to also increase the blood pressure because we have a fixed volume inside of this system. This is another reason why atherosclerosis is such a serious issue. As we start to deposit plaques on the inside of these vessels, that's going to drastically take up valuable volume for our blood to be sitting within. And so that increases our overall volume of things found in this closed transport system and drastically will increase blood pressure. So as I have already mentioned, arteries are going to take blood out to the tissues. Now, one thing that we've just learned about arteries is that there's going to be elastic tissue as well as smooth muscle that's found in our arteries. Since the heart is directly pumping into the arteries, we have to have a large amount of elastic tissue to allow for the stretch of the vessel walls. As the blood is constantly pumping blood directly into the vessels, our arteries have to be able to withstand high pressures. The more elastic tissue in the vessel walls, the greater the amount of stretch allowed by our arteries, and the more that they can absorb the forces exerted by blood inside of them. We will also see the elastic tissue in the arteries facilitating the production of laminar flow. As the heart is constantly pumping into the vessels, the flow of blood is somewhat disturbed, as each individual heartbeat is separate from one another. But due to the stretch on the vessel walls, as the vessel wall continually squeezes down on the blood inside of the vessels, that's going to produce a more smooth flow of blood through our vessels. The other big thing that we'll see in our arteries is smooth muscle, and this is very important that we understand that this smooth muscle is circularly arranged. This allows for the tunica media of our arteries to constrict and close off the lumen to narrow the diameter. This can help to regulate blood flow to certain areas of the body. If we vasodilate and allow for our vessels to open up, that is going to facilitate more blood going into that set of vessels. But if we close and vasoconstrict, that's going to stop blood flow going to a particular area. The key with arteries is that the tunica media is going to be much, much thicker than it is in the veins. Now, there are two major types of arteries. Now, we don't want to overthink these. These are very straightforward. We have muscular arteries and we have elastic arteries. And we can see, if we come down to the second bullet point, the muscular arteries are going to be more prominently containing smooth muscle and will have fewer elastic fibers. And the elastic arteries are going to contain more elastic fibers, but not going to have as much smooth muscle. So don't overthink these. These are exactly as they sound. The elastic arteries are in fact the larger of the arteries, such as the aorta and the branches that come from it, such as the brachiocephalic trunk, the left subclavian artery, and the left common carotid artery. All of those larger vessels are going to be right next to the heart and receiving blood flow. Therefore, we need to have more elastic tissue in those vessels so that we can withstand the greater pressures that are coming out of the heart. The elastic arteries will eventually get smaller and smaller and start to have more muscle tissue found within them, and those are the muscular arteries, which are our more medium-sized arteries compared to the elastic arteries. We call the muscular arteries the distributing arteries because it is these vessels that will specifically direct blood flow to and from other places. Now, it is these muscular arteries that are going to use that smooth muscle to control what areas of the body receive blood and which do not. It is in these muscular arteries that we have vasoconstriction and vasodilation. Now, we know that the arterial tree is going to go from large to small. As those muscular arteries continue to branch, they get smaller and smaller and smaller until they are classified as arterioles. Arterioles are the smallest of all the arteries. These are the last vessels of the arterial tree that will eventually flow into the capillary beds, and these will also have a tunica media that will facilitate constriction and dilation to regulate blood flow. Now, the tunica media in the arterioles is much, much thinner than in arteries, but regardless, arterioles will still facilitate the process of vasoconstriction and vasodilation.